really see from here, but it makes no sense because it has no tube and it's on a rock. So we don't even, we're not even sure what phylum, what, excuse me, what order or class, I guess, of Cnidarian. Yeah, let's get some good close-ups of this. Expect it to swim away when you try to grab it.
There's a seriantid on the other side of the rock, that face that you can't see. There should be a tube.
That's next zoom.
And when we did a cruise back in 2004 in the Aleutians, or 2005, one of the two, we did see a seriantid tube anemone on a rock. Its tube was glued to the rock, and we were able to collect it because the rock was loose, so we could pick up the entire rock with it. But otherwise, I think it would be really hard to try to grab this thing.
Well, they just got to prepare for it to swim. We almost got it the first time, and it does not have a tube stock. Scott, I can absolutely assure you it has no tube. But it's got that inner oral tentacles all around it, but you can't really see at the moment.
Can you let me go? Let me know if I'm getting into trouble with that swing arm.
Yeah, absolutely. Looks fine right now.
Yeah, Tina is saying is okay, they can live without tubes, so thank you very much. Tina Molotova is now wide awake, even though it's late in Moscow. Probably her eyes popped wide open when she saw this
What
thing.
time are you calling the move, Sean?
So thank you. Tina's from the Shirshov Institute in Moscow.
Only 2 minutes ago.
Okay.
Yeah, so you're not going to feel
It does
anything
have that
for
inner row
maybe another
of
2 minutes.
Yeah, so we're gonna try
Yeah,
this.
if we can look it down the center of it, that'd be great.
Okay, I'm still
Yeah.
Just gonna
rotating around.
All right,
turn
now,
my head a
pilot,
bit
you
toward you.
careful with your light pole now.
Watch your tether, because you're gonna start tugging me.
Yep, I did.
We're not gonna be moving for another
Come
couple
out,
minutes. Yeah, you need to come to
That's the end
starboard.
of the tether.
Come to starboard. Yeah. We'll be there soon enough. Just give it a couple minutes.
Yeah, pilot, if you go back to that position once you have tether,
I'm trying to figure out
you might
how
wanna
you're
pick
going
up
to do
your
this, pilot.
port
Because he's
lower.
on the left side of the
'Cause you
boulder
got-
and the arm's on
I
the
mean, it
right
was still
side
safe,
of the
but-
ROV.
Yeah, I think we'll figure it out when we get a little more tether. We can get closer.
Sure.
But I think we can do it by setting up sideways and looking directly at it.
Okay, thank you. This is one of the strangest mysteries that we've seen in the 3 years of the Capstone expedition. That's why this is worth a little bit of time here. I mean, when you can't even go to class or order or something like that, then you really don't know what you're looking at. Yes. Thank you.
So we'll go ahead and bring in that swing arm now.
Okay.
Yeah, it's a good preemptive thought.
There we go.
Also, could I make just a suggestion, even though this is your specialty? If you had the box open and ready to go, you can almost, it was Sean, wasn't it? Are you over there in nav position,
Yes.
Sean? Was it, you were the one who was trying to get this, right?
Video call again.
When was it?
We've got a little
This
more
was
tether
like
now,
the
Dave.
first dive, I think. Remember the big, huge anemone-like thing that swam away? And then,
I don't think so.
I'm trying to think who was on the stick at that time. It was Chris Ritter then, apologize.
So the
Annie
thing
White
about
just
that
told me who it
is
was.
that,
So
it's kind
you
of on
might
the
even
slope
have to
here.
corral
You're
it
not
into
gonna
the
be able to sit
box.
down
That's
in
why I'm
front of it if you
suggesting
got
if
the
the box
drawer
was
open.
open, if it starts to swim.
In some cases, I would agree here. The problem is if we have the drawer open, that makes it a lot more difficult to set down
Okay.
for a really stable grab.
Okay, yeah. all right,
Otherwise,
okay.
I would certainly like to, and to get it in there as quickly as possible.
Okay. And also, Chris tried to grab it around the thicker base there, and then it just took off to the left, if you will. So, if you can, I'm wondering whether even if you could try to grab it head, you know what I mean, sideways, so you prevent it from swimming off.
We'll do our best here.
Yep.
We'll think about this before we try to grab it, but.
All right, video, come on back out, and we'll start to get set up here.
Okay, I'm just going to grab a quick specimen view.
Oh, sure.
Well, actually, at what point did it start to swim away? Was it when it first felt contact with the craft, or could it kind of feel it?
No,
when it
first felt the manipulator touch it.
Okay, copy.
Okay, full wide.
All right.
So what I'd recommend. Actually, why don't you tell me what you're thinking, Dave?
Well, this is a nice place to sit down, but I have the rock in the way, so I'm thinking I go over to the left, turn almost 90 degrees to starboard, and possibly come down over its body like that. Sure. Because then it can't swim out, at least. What do you think?
Yeah, I'm not sure you need to go like 90 degrees from it, but kind of at an angle. Yeah, just when you're only going to be able to get the one toe in.
Yeah.
Looking at the event log.
Unless
Sorry,
you
I've been
kinda
so
try to
focused on the
put your
video,
other
I haven't
starboard
noticed
toe on this
the
other side of
event
that
log.
rock.
Maybe.
But I don't know if bumping the rock would cause it.
Scott, we'll
Starboard
never
everything.
find one on a rock that. It's just a really big beast. I don't think it'll work like that, but you'll see
Should
when
this
we
stop
try
to
to
make sure?
grab it what it's going to do.
Yeah, it stopped 2-3 minutes ago.
Okay, perfect.
So you might have a minute or so of swing, but besides that, you're good.
Good.
Yeah, I think Tino was not on, but Asako was, and Asako remembers exactly what it did, and it surprised the heck out of all of us.
Co-pilot, can you joy lock? Never mind.
So,
It's pretty far down. isn't it?
yeah. A lot less maneuverability with the arm if you're high up like that.
So, one step further. Back to the front. I think I have to go up there. Where it gets bad. Pull back and get a better look.
Yep. Just take your time. It's fine to experiment a little bit. Try to find your best footing.
It's a difficult spot. So you think perching above it is?
Well,
It's not-
it just you
I
might be
know
able to
it's-
set your port toe down on the rock on the left side, you know. And the nice thing about this is you can just come in from the top so you wouldn't have to reach down too far. But
Okay.
then, you know, the further above it you are, the less maneuverability you have, you know,
Right.
to come on top of it. So it's the fewer angles, the smaller angle at which you can grab it.
Right. But coming around that way is also a problem if
I
you set
don't.
further down.
Yeah, I'm not sure. I'm not sure if I'd approach it from that angle, because
You think more head on?
I think coming at it from the starboard side, as much as possible, because even then it's, I mean, I understand your strategy with keeping it kind of boxed in, so we can't swim away, but, you know, if you're kind of angling the grippers, the claws toward the rock, you know, you can
Right.
try to get it trapped there.
Okay, we'll try this.
I'm going to see if, well, you generally want to remain a little bit quiet because this is a difficult collection, and so that's why we're being real quiet here while they concentrate and discuss how to do this. And so as soon as we got this in the box, I'll relay some of the chat room comments.
You sure like me? Sure. Perfect. Heading back.
So let's say you go for the grab here. What is your strategy with orienting the arm? This is important to think about before you go for it.
I think I have to go back past it and come back in with the wrist about that angle. Grab it around the base with the wrist facing toward it.
And what are you thinking? The grip force, and how are you gonna pick it off the rock, and where are you gonna put it?
It's going to go port side. Probably 3 grip force. I will come in like this, grab it around the base, pull it out.
Okay. And how are you gonna deal with the grip?
What do you mean?
Are you just gonna grab it?
I think so. I think I want to do it fast. Right?
Okay. But do you wanna clinch it too tight?
No.
Right. So you're gonna try to feather it in.
Okay.
And I think it's worth, you know, before you get too close to it,
Yeah, do a couple.
practice feathers and try to even come in. So you just have to get a little more and close it around the jaws.
Okay.
And then what's going to happen once you have it in the jaws? What are we going to need to do?
I'm going to need to lift off.
Yep. So you'll have to lift off, and what are the mechanisms through which you're going to do that?
We'll come out of joy lock, probably leave auto heading.
Okay.
And come up and back.
Okay.
Lock in depth.
Okay. Yeah, and with the auto depth, what I'd recommend, you get up off the bottom, you lock in auto depth, and then you give the set point a little bump up, such that the thrusters will keep trying to push you up, and you'll have a little bit of momentum to keep you up, instead of if you have momentum upward and then you lock it in, and then
Right,
it has to fight to come back down, and that can be precarious. So,
Okay.
yeah, I think we have a plan. So when you're ready to execute,
All right.
we
Prep
can
starboard bio box.
Yep.
And spin up.
starboard or port.
I'm sorry, port.
Okay.
Definitely port. And are we spun up?
Hydraulics are ready to go.
Great.
Grid force 3.
Do you want me to take your camera?
Sure.
Okay.
You can take both, actually.
Okay. You want me to put up Mini Zeus?
Yes, please.
Okay. Sean, if you can help me keep an eye on the Sirius camera while we're distracted here.
Yeah, copy that. I'm on it.
So, bring. Not getting this rotation. Am I in continuous? No. Oh, I'm against the limit. Okay. Struggling a little bit with the wrist here. So Copilot, I'm hitting. I want to have some range of motion on the wrist rotation.
Yeah, so when you put it in
I'm
continuous,
putting in continuous going
and you can
past
go
it.
as far as you want. Yeah,
But then when I come back out,
you have to bring it back. It has limits in manual rotation. So if you're hitting a
that's
stop,
my
you gotta
stop.
you gotta
I go to
spin it,
continuous.
or just bring it back. Yeah, bring it back the other way until there you go, until you center it out.
Right, but it's less than one full rotation. Okay, there we go.
And watch how close you are to the Mini Zeus here
See that.
with your wrist.
Can you come up on main?
Yep, coming up.
Still struggling with the rotation a little bit. So that's a stop.
Yep. So you can rotate it and then index out, bring it back, index in, keep rotating until you've centered out the rotation.
Okay, center the manual range.
Yes.
Go to continuous, get it about where I want.
Yes.
Then, nope.
So don't even put it in continuous. Just keep indexing in and out and rotating it manually until you've found the center of the rotation.
Right, that's one end. That's the other.
So watch your wrist. You should bring the arm down a little bit. You're really close to the Mini Zeus. Yeah.
That's the center.
Okay.
And then I want to use continuous to move the center to where I want it. And now I can't rotate left.
I'm not sure if the continuous is going to fix the set point. I always do it manually with indexing in and out. I don't even touch continuous when I hit the stop. I index in, I rotate it manually, index out, bring the controller back, index back in.
That's what I'm doing.
And it's
I
not-
can't go any further.
I'm not sure. That's something we'll have to look at later.
And I can go further. See, okay. That's a stop. Bring it back. Can't go further. And I can go further with continuous. All right. I will just deal with it. does my alignment look, Copilot?
You can look in that manipulator and see that you're a little far forward of it.
Okay.
Just one more note I'm going to say before you go for it.
Yep.
It might get away, and that's okay. But just nice, slow, controlled movements. It starts to swim. Let it go. You know.
Okay.
I think you're still, yeah. If you look in your
Yeah, I see.
manipulator camera, it looks like you're pretty far forward of it still. Yeah. Watch your claws, your outside claws are about to hit it. Alright.
Okay.
Well, there goes the stock. Got some tentacles.
So we're going to back up, come off. Outer depth, bumping it up.
I would go ahead and bump maybe auto XY as well. That'll keep you from going too far with the current.
Okay, can you open that box?
Yeah, you got auto altitude.
Whoops.
Yep. There we go. Drawer is opening. With port bio box open.
Great.
Tilting down on Mini Zeus. So once you get it positioned in the box, I want you to index out. And then give yourself a bump upward on the auto depth.
Having some control problems beyond the wrist.
That's fine. And just give yourself a tap up so that the water gets forced down into the box. Looks good.
Okay. Ready?
Go ahead. Oh, it's stuck.
Stuck.
It's alright. Nice and slow. Get it in the box. Everything's safe. Just nice controlled motions. try rotating your wrist, jiggling it back and forth. It's alright, nice and slow. You could try kind of lightly scraping it off on the side. But just make sure you don't put too much pressure on the box.
Okay.
You're almost there.
Can you see where it's stuck?
You could try rotating the wrist. I can't quite see. There you go. Oh, is that it?
That's
all right. Clear the arm. Arm is clear.
Arm is clear.
Retracting. Okay. Drawer retracted. Pins retracted.
Okay, can you follow me to stow?
You got it.
Actually, I'd like to run the arm through some movements. I was having a little trouble there. I just want to make sure everything's okay. Yeah, there's a stop.
Something I'll have to play with later.
Yep.
I'm not sure.
Right, they just, let me update Chris for a moment.
Okay. Bring it up to stow.
So this is Chris Ma from the National Museum of Natural History. I'm back from lunch and ready to
Nice job,
ready
Dave.
and raring to go.
You got it.
So if we could head up at your convenience.
All right, taking my camera back.
All right. I'll give you Mini Zeus back as well. Sure. So I think we're facing 065 right now. I think 070 is the last move, and that looked pretty good
I'm
to me.
centering up.
Yeah, that's correct. 070.
Yep.
Last move.
So I'm looking there now.
Centering up.
Kind of zigzag back and forth, and maybe we'll get a move in here pretty soon.
Yeah, and remember the lower port was stored.
That's right.
You could bring that back out.
We'll bring it back out. Thanks, Sean.
Check your tether right under you.
Yeah, hold on 1 second. Sure. I'm pretty high up, so I think we're pretty safe. All right.
Yep, it's fine.
That's good. Yeah, 18 meters above you, so.
Yep, I was just looking at this.
Good.
I wanted to make sure it was open.
Do we want to look at this brown thing
Yes,
coming
I'm
into the
getting set
center?
up on that. Let's turn lasers on,
You got it.
and you can come in.
Looks like a dead sponge skeleton. And what's this below it? There's a dark...
Below?
Yeah, there's a flat, dark structure that's just...
Oh.
Yeah, let's zoom in on that.
Like another 4M possibly, Chris.
Oh, is it a 4am? Ah, okay. Okay, we can get a... If we don't get a establishing shot, we can just move on.
Okay, video, please come back out. And we'll continue up.
Thank you, sir. Much appreciated.
I take that back. Snap in on this.
Nice feather star. Echinoderm filter feeding. Uses its arms to pluck organic particles out of the water. Undoubtedly one of the many that we'll see today. We're good. Thank you.
Okay.
Can we hold just 1 second?
Sure.
Looks like it's got some nice eggs or something on the pinnules near the mouth. Fecund crinoid.
Okay, video coming out.
All right.
Full specimen view.
Always was a little closer.
Thank you very much.
Come on out. Now let's make a note to not open starboard port bio box.
Note already made.
Great. Something in the water column?
Yeah, I see it.
That. Go.
Oh, neat. Looks like a little radiolarian or some kind of protist. Oh, wow. That's cool.
Nav, can you watch my swing on?
It
Yeah,
looks
you're,
like
you're
a
good.
satellite.
Yeah. Little Sputnik.
Let's let it go.
There's a holothurian up near the top, a little bit to the left
see
of
it.
the screen.
What's that purple thing up ahead of us? Is that just a dead sponge stock, or?
I think it's about to take off. Let's come in.
What is it? Oh, it's a sea cucumber. Big sea cucumber, I'm guessing. What are the lasers on that?
Come out a little bit for lasers.
wow, it's 10 centimeters long. So this is some kind of a lasiopod sea cucumber, maybe in the Elpidiidae. Notice it's got the long tentacles coming off of it. The edge on the left side is where the head is located, and it's got its feeding tentacles, like it's prepared to, and you can see some of the sediment that it's ingesting. It will devour the goo that's sort of rested or floated down on a lot of the sediment, and it swallows it. It goes into the intestine, which you can see very conveniently through its transparent body wall. These then can essentially cycle the sediment through their body, and they deposit sort of the clean sediment cast afterwards out of the end. So sort of like an
Should be
earthworm.
stable. You can come in.
This one's quite large, so this is a productive lifestyle for some species. It's also got some interesting things
Sean?
growing on the
Let's go ahead and put a 20 meter move in.
tentacles.
Copy.
I'm not sure if that's
copy that.
epizoic or if that's just part of the animal, but these are important for bottom ecosystems in the deep because they help cycle nutrients and other items, as well as clean the sediment up
Go ahead now.
or aerate the sediment for different kinds of animals that live in the substrate. Great, we can move on. Thank you.
20 meters at 070.
Video just wants to
Chris,
stay
do
on
you
this a
know,
bit.
has anyone hypothesized what the function of those elongated podia would be? I mean, they're not like the sails of the swimming ones. I'm not sure what
That is a good question, Scott. I honestly don't know because, I mean, I think some people have thought that they might be sensory in some way,
ROV nav bridge, move of 20
maybe
meters,
to
running 070.
detect water current, or maybe even to detect how the goo that they like to eat is coming down. Dave Possum, the curator at the Smithsonian for sea cucumbers, has mentioned to me that you'll often see these, if you see them in a massive field, they're all oriented in the same direction, sort of directed towards where the food is coming from, and so possibly they're able
Chris,
to...
sorry to interrupt.
Yeah.
I think there's a little white branching structure just near the head end of this thing that might be a bryozoan just past it, so if you're still there.
Yeah, I think those were little forams and things that we were seeing earlier. Those little branched...
Okay, I
yeah.
thought this was something larger.
Oh no, it's well. Let's pull out if we
It's
can.
behind the hood right now.
Video, if you're happy, you can.
Is he talking about the white structures growing on the holothurian?
No, I don't
I'm not
think so.
sure.
He's talking about something on the rock.
Find it on the rock, and I don't think it's a foram. I think it's a little bryozoan,
Oh.
and it's partially obstructed by the oral hood.
Oh, okay.
But when we first zoomed in, you could see it very clearly. So we
For
can
wide-
look back at the video later.
Yeah, that's great. We can move on, pilot. Thank you.
Okay.
Nothing makes Wednesday better than a 10 centimeter long holothurian.
I can't think of anything.
Unless it's a 20 centimeter starfish eating that holothurian.
All right, it's another 3 minutes or so. We'll start feeling a move
All right.
upslope. But until then, if you kind of want to come to starboard and pan across.
Got a swimmer here. Come in. Let's see if we can get this.
Blowing them
Switch to
around.
This one. All right, you can come back out.
Actually, yeah, it kind of feels like I'm seeing that move already. So,
All right.
Let's take a quick look at that branching structure over to the right on the boulder there. Looks like a toothbrush.
Oh, okay.
Thank you.
Right above the lasers?
Yeah.
Okay, come in.
I don't see it.
Right between the lasers now.
Yeah.
Oh, okay, I got it.
Coming down. Go.
Looks like a little tube worm of some kind. And let's pan left also. Our intrepid. We could move to the
Let me
left.
just get this. Okay, we got it.
ROV bridge, move
Oh,
complete.
when you're done, of course. Thank you.
How far left?
Looks like a
It was only a
polychaete
20 meter
sandworm
move,
with
so
some barnacles
you're
growing
fine.
on the tube,
Yeah.
perhaps.
Ah, here we go.
Oh, okay.
Oh, is that a? That's not a black coral, is it? Or is that a juvenile? Yeah, we could zoom in on that. As,
You can take some more.
so if our shoreside talent could chime in on this, I think that's a. Is that a black coral?
Yes, well done, Chris Moss. That looks like maybe a very small Bassopathes black coral.
I must relinquish responsibility to Nicola, our intern, who is
Do you have any
far more
video?
sharpshooter of an eye
That's
than I
my
did
son.
with this one.
There's something near the top of it.
Yeah,
Question is, what is that little orange thing that is on one of the branches?
that's a good question. Lots of those today. Might be, I don't know, a parasite of some kind?
I'd love to get closer, but this thing is tiny.
Yeah. If this is the I think we got enough of a shot of the front. We can move on. I think that's enough for them to see, at least for now. We might even see a bigger one later.
Okay, video whenever you're happy. And we'll hit the base of it with the lasers. If you come out a little more.
So, the name of our expedition is the Lima o Kamoana, which means Partnership for the Ocean. And right now, there's
Maybe
nothing that
he ranged down
demonstrates
on V2
that
sonar.
more with regards
So
to our
about 30
research
meters
than the collaborative
forward.
efforts we have between people such as our junior scientists,
There you go.
such as Nicola Rodriguez, who helped spot that black coral, our pilots,
Hey, Sean.
our
let's,
videographers, and of course my
actually,
colleague,
right. Why
who
don't you come up,
is
We're
switching
gonna
out
do a
with
steeper
lunch with me
feature.
right now. I've already gone. He's out. He's already down there.
Should I not
So
stop for it?
Yeah,
big efforts like
come
this
up.
don't happen by individuals, but a team.
I think you need to come up vertically. See, it's a
Oh,
little,
yep. Oh, wow.
little sharp here.
It's pretty steep.
Copy that. So we should have maybe a minute or two swing left.
Let me just
Wow.
locate
Yeah.
the top of this.
Punch it up.
Yikes!
Then we can take a look at it.
Yeah, I was seeing over it, but didn't want you to get caught down there.
Sure.
So we're looking at a large magma boulder thing,
All right, that's the top.
Okay.
and we're coming
Okay.
up over the top.
So why don't
Wow.
we can wait for Sirius to settle out, and you might be able to get back down there.
This, I believe, is where the crest, the ridge, or something, or rather the ridge begins.
So yeah, if you want to come to your port, I'm going to bring
Yep.
my head back to
Coming to port.
070 or so.
Video switching out.
Thank you, Emily.
And Chris, if I recall, we believe we are on the lee side of this mini ridge that we're climbing, and so when we get to the top, perhaps the currents will be stronger. Is that correct?
I think so. Yeah. Where are we? We're right. Yeah, we're right on the contour line.
How much left in the move? Edge?
Move ended. I
Yeah,
would
yeah,
bet
yeah.
Cirrus is done swinging.
Okay.
It's been almost 10 minutes. Exactly.
Yeah, so now, of course,
20
it's
meters,
right
yeah.
there. Yeah.
Well, I'm looking down at you. I'll keep the camera at like 80 degrees. and
Okay.
I'll keep an eye out. But I think you can explore a little bit if you want.
All right.
So what's the current like here? Is it have we
There's very
suddenly
little
accelerated?
right here. I think there's a little bit in my face, which is down the slope
Okay.
out of the east.
How you doing,
but
Roland?
not much.
Okay.
We have just a
Doing very well.
minute or two here that I can poke my head back down the wall before we
Yes.
need to
If
get out of here.
you have a moment,
So we'll do
some
that
of these
quickly.
little white. Oh, there's a coral or something like it. That branching structure right in the
Run
middle.
in on this video.
Yeah.
Ready when you are.
Come in. Just do
Oh,
a
this is our
quick
first
one
coral of the
below
day,
here.
I think.
It takes
And
a
I think it starts with a chryso. What do you think, Scott?
I would agree with you, Chris. I would dispute it's the first coral of the day if you include sea pens and black corals. But it's the first octocoral that lives on the hard substrate, so I'll agree with you there. And it is a member of the genus Chrysogorgia in the family Chrysogorgiidae, which are the golden corals, and it's very clear there. You can see that golden color through the thin tissue. And this is a really nice angle to see what Les Watling has said several times about how it is you characterize these. And we're getting to a view now where you're almost seeing 3 tunnels going down from the top of this colony and through it.
Oh, wow. Yeah, that's right. I can see that.
Branches, as it grows, is perfectly aligned with ones below it. And one of the ways we determine the taxonomy is how many revolutions
get tight
of
on
the
the crabs.
main axis do you have to go
Squat
to
lobsters.
for that branch to be
Talk about the
perfectly
commensals.
aligned over
Not
the
gonna
lower
be
one.
stable, but
And I
we can
wonder
confirm.
if this is Chrysogorgia tricollis, based on those 3 sort of openings that.
Oh, is that where
are
the name
framed by
comes from?
branches.
Interesting. I noticed, too, all of the polyps are oriented in the same direction. That's always fascinating to me.
Yeah, I think that's just showing you the direction of the currents right now. They're pretty delicate polyps, and they can be bent over. It also looks like some of these polyps have the saddlebags that Les was referring to yesterday. So you see where the polyp is connected to the skeleton. Many of them are much brighter white, and that's basically gonadal material, probably eggs.
Oh, really?
And there's such a high production that the bottom of that polyp sort of extends on both sides over the skeleton, like the saddlebags on a saddle of a horse or on a bicycle.
Oh, neat. Okay, well, I consider myself edumacated. Those are reproductive.
Can you
Cool.
tell us anything about those 2 commensals on it? Or what a commensal is?
Yeah, absolutely. And so in the branches there, sorry, my video's frozen. Okay, sorry. So yes, in the branches of this Chrysogorgia, you see that there are 2 squat lobsters, and this is not unusual for many species of Chrysogorgia and other octocorals, and they probably have a pretty tight host association, meaning that this species of squat lobster may be very specific to living in just Chrysogorgia, for example, and it's entirely likely that we may have a male and a female. We have seen that on past expeditions when we've made collections, or maybe it's 2 females, both of whom have eggs, and they're basically probably using that colony as a perch and as protection. And so this is one of the reasons why we find exploring these deep-sea coral communities is valuable, because the animals themselves create habitat for other animals to associate. So it's kind of like birds living in the branches of trees in the forest. That's essentially what those shrimp are, what those squat lobsters are doing.
Great, thank you, Scott.
Sure is. Leave it to Roland. We'll get you what you
A
need.
little bit of fishing.
Well, there hasn't been much to see. I think that's the first associate we've seen.
Oh, there's another one up on the top there in that little sort of
Straight above lasers.
Oh, yep, I see that.
uneven area. Yeah.
All right, video, come on quickly. I think it's the same.
Yeah, it's the same one. But here is another Chrysogorgia with its 2 squat lobster commensals living happily.
So we were just talking about associates, so perhaps this is another good example. You don't see any of these squat lobsters on the rock around these corals. We're only seeing them on these corals. And we didn't see it on the black coral or the sea pens we saw earlier. So that's an indication that at least some of these, what we call associates, invertebrates that are living in association with a coral or a sponge, have a preference to live in certain kinds of species of coral or certain species of sponge.
Video's
And
clear.
it suggests that the larvae of these animals are able to detect using chemicals in the water that are given off by the coral, so they can detect them, land on them, and then perhaps grow there. Alternatively, as juveniles, perhaps they're moving around the rock until they encounter this colony and then move up to it and stay with it.
Continue down laterally
It's
for
an
a little
interesting
bit.
question, and one that requires further study about how long is
I wouldn't
this
go too far down, but
relationship
I think
in
you
the
have a little bit of
lifetime,
freedom
for
here.
example, of the coral or the crab. We certainly know that hopefully today we'll get to see some of the Metallogorgia octocorals. We know that the ophiuroid brittle star that lives with those stays with it its entire life.
Yes, and when we see these in the wild, or when we've seen them
No
in the
straight
past
center
few
if
days,
that's a small
it's
whip
really
or just a
strange to see how they always have an associate
illusion.
present. Very
Not
rarely
seeing
do you see
video.
one without. Rarely do you see one without the matching set, as it were.
Right hand side of the screen. I think it's an
fascinating.
illusion.
I just see sediments and cracks. Yeah. Alright, I'm going to come up over here. And we'll go on top
There's
of it.
some new
Oh,
guy
there's
at the
something
top
up on
left.
the top there. Oh, maybe it's just a rock.
Are we running with lasers or?
We had just turned him off for that
Oh, okay.
associate shot.
All right, and we're up over that little wall.
Lovely. So we're tracking over what looks like a wondrous field of
Went off screen up left, maybe.
cooled and solidified post-lava rock.
Sponge upper right.
I see that sponge. I'll go for that.
I'm gonna sit down super close to it.
Oh, and there's a nice euplectellid over on the right there. It's a glass sponge coming up on the upper right-hand corner.
It's like a dead attachment point passing off the bottom of the screen now.
There's also a stick on the rock to the upper left.
Yep, I see that. I'm going to go for the sponge first.
There's a chance we can look down inside.
So these are
Maybe.
animals made of glass, and we're seeing as we draw in, you'll see that their body structure is quite elaborate.
So I'm back in
Welcome
time
back,
for a
Chris.
sponge.
I had one specially made for you.
Thanks.
Get a little bit closer before I sit down.
Euplectellid, if I'm not mistaken.
Yep, you are not mistaken. This is
Okay. Video, come on in.
So sponges are in many ways like the corals that we've been seeing. They provide habitats for a lot of different animals. In the case of some of these glass sponges, there are actually some fairly elaborate behavioral stories that are recorded around them. In Japan, there are some shallow water glass sponges that
Sure, come on
have
out.
shrimp
Video?
that
Let's
enter
get
into
a
them,
wide
into
shot
the internal
first.
chambers as juveniles, and when they mate, they stay together, trapped within the glass sponge cavity because they've become too big to leave.
Co-pilot, that's probably enough with lasers.
This is a euplectellid vase, as you said, and it could be one of two. Could be one of several actually. could be a Dictyolis. It looks more Regadrella to me, so I think I'm going to guess that for the annotation log.
Regadrella.
Yeah, so these are sort of classic glass vase sponges, and this one's got a what's called a sieve plate over the opening
see
on
the
top
top?
of the hole, the main opening, kind of like a bottle, where the water comes in the sides and then it's sent out through the top. And this is one of these sponges where I was mentioning the other day that they may be taking advantage of a Venturi effect. This morphology may actually be creating a Venturi effect where water passing over that opening draws water through the sponge at a more effective rate for filtering.
I always love the biophysics of filter feeding animals.
We're probably good on this one. Our video is good because this is a pretty common sponge.
Okay.
We haven't seen it today, though, have we?
Yeah, but it's recognizable, so thank you. And we'd like to get up near that crest if we could, please, and get into a really nice community with any luck.
All right. Should we get another 20-meter move in?
Let's do it.
Yeah, see cameras at 5.5. Still want to go at 070?
That's fine.
We can adjust
Maybe a
maybe
little more to port.
More to port, yeah, that's what I was thinking.
copy. How's
This is Bruce again.
at 6.5?
Noticed that
6560?
saw a couple of fish early on when we were in the
060?
sediment.
Copy. 20 meters,
Now that we're moving
060.
up into a rocky area. It's been a long time since we've seen any fish, so it'll be interesting to see what occurs later on.
Okay.
Yeah, thank you, Bruce. I think we're all kind of eager to see what the top of the seamount looks like. After yesterday's find, we started out. We saw some stuff yesterday in the bottom, but when we got on top, that was the glory hole right there. So I don't know if this can be repeated on this dive. It's, but it's certainly got
confirmed bailout.
people curious.
5 degrees.
Yes, sir. That's correct.
All right, thank you. Coming up.
Yeah, I was just telling our the executive officer of the Okeanos Explorer that there's nothing quite like
screen and the tight
sort of having the camera roll up on a ridge or something, and then automatically having all of these sponges just in all of their brilliance to show up suddenly like that. The ensemble appearance is really quite stunning.
So we still have a lot of loose rocks here, too. I was expecting it to. I mean, we're fairly close to this little saddle,
rock.
so it's not surprising because rocks could have been moving down slope from the northwest too.
Snap on it. Come in, video.
Dead sponge base. It's probably the base of one of those base sponges too, because some of the euplectellid vases kind of have some finger-like structures.
No, it's like it's got a little
worm tube on it.
Yeah.
Pulling out.
Okay. Co-pilot lasers on, please.
You got it. Lasers on.
Taz, before I actually
Something under that rock to the right.
switch my switch on to active audio, I did sort of take a quick glance through C-Scribe to see what you guys had seen, and you saw a bunch of chrysogorges that look like along this wall.
Oh yeah, there were some present on the vertical surface. Some Chrysogorgia tricollis, yes, with some spot lobsters. Scott gave us a very nice presentation on that.